Thank you guys so much for the invitation to be here. I've had a really wonderful time over the last few days. The breadth of talks has been really refreshing, and I've gotten to learn so many new things, so thank you so much. So for those of you who don't know me, I'm Emily. I'm from Penn State, and in my lab, we study the interaction between humans and our microbiomes. And the two kind of main overarching questions that motivate our work are, number one, what factors determine microbiome composition in a host? And then, does that composition matter, either for health or for evolution? And we come at this from kind of a unique angle, which is that I am trained as both a microbiologist, but also as a human geneticist. And what I really try to do is I try to bridge those two fields together. I try to take techniques and ideas from human genetics and bring it to the microbiome world. And I try to have a very microbially influenced understanding of what's going on in human microbiomes, not just as a human geneticist. So the main kind of techniques that we use in the lab are things like microbial ecology, genomics, and human genetics techniques, but we also throw some statistical genetics and PopGen and some systems biology in there as well. So just very briefly to give you an overview of the kind of things that we think about and we're working on, our kind of main area of focus is this direct intersection between host genetics and the microbiome. And I'm going to tell you one story from that area today. We're also really interested in expanding our understanding of what microbiomes look like all over the globe. So the other day, we heard a great talk about how in the human genetics world, there's a potpourri of studies that are done in certain populations, with many more being done in individuals of European ancestry. The exact same thing is happening in the microbiome world, and in some ways even worse than in the human genetics world right now. So we're really trying to expand what has been done with global microbiomes. We're also really interested in the evolutionary consequences of having a microbiome, and we're doing this, what we can do in humans, and also turning to Drosophila as a model organism. And then finally, like most people here, we do a bunch of methods development. We often have questions where no method exists to be able to study that question, so we develop things both in the wet lab and computationally to answer those questions. So like I said, I'm going to tell you one story from kind of this host genetic element today, and it's actually going to be kind of more on the genetic side of things and not the microbiome side of things, but don't worry, the microbiome is in there. And so yes, we're going to be talking about using a microbiome as a proxy for environment in a GWAS. So to motivate this, one of the major questions in all of biology, right, is what contributes to inter-individual variation in a trait. And this variation is incredibly important because this is what selection acts on, and it is also, makes some individuals more prone to disease. So one kind of easy to visualize phenotype that has a lot of variation is height, of course, right? Some individuals in the population are short, like myself. Some of you guys are really tall, and then most of you are somewhere in the middle. And oftentimes, we're really interested in understanding why this is the case. So as geneticists, we're really interested, of course, in the underlying genetic architecture of these complex traits. And in complex traits, right, it's usually not a gene that drives this stuff, but it's many, many, many variants all over the genome, most of which have very small effect sizes. In addition to genetics, of course, environmental influences also influence many complex traits. So for something like height, things like nutrition, the medications you take, exposure to a toxic chemical spill when you're young, something like that, those can all influence how tall you end up being as an adult, right? And one kind of emerging environmental factor that, of course, I'm particularly interested in is the microbiome. So the microbiome in humans is a largely environmental factor that we carry with us from the time we are born to the time that we die. The back of the envelope calculation is that there's at least a 1-to-1 ratio of microbial cells to human cells in the human body. And within those microbial cells, there's an estimated 100 times the gene content as in the human genome. So if we're thinking about the metabolites and other molecules that are circulating in our bloodstreams, actually, the majority of those are microbially derived and not necessarily something we generate from our own genes. The composition of the human microbiome, at least in the gut, which is what I'll focus on today, is determined by a lot of different factors, most of which are environmental. So the diet that you eat, the drugs you take, your exposures, and also to some extent genetic. And we know that this microbiome can affect host physiology. We know that they help break down our food and the drugs that we take. We know about those microbial compounds that are circulating in our bloodstream that we interact with. And of course, the microbiome trains our immune system. So depending on the exposures you have when you are young and when you are developing, that can determine immune signatures throughout a lifetime. So with all of this said, there are two kind of big questions I want to walk you through today. The first thing I wanted to ask is kind of like the duality in my brain anyway, of understanding complex traits. How much of this is due to genetics, and how much of this might be due or explained by the microbiome? And the second question I wanted to ask is, can we actually use the microbiome to inform our genetic studies? So to do this kind of work, we need kind of a unicorn data set. So I'm not interested in one disease necessarily. I really want to understand patterns across complex diseases. So to do this type of work, I need a bunch of different phenotypes, I need microbiome data, and I need host genetic data. And this sounds like a lot of data, but this exists, right? In the human genetic world, we have these giant biobanks like UK Biobank and others where there's a ton of genetic data and a ton of disease and other phenotype data. However, in those biobanks, we're lacking microbiome data typically. In the microbiome world, we have also a bunch of big consortiums that have come out with microbiome data sets where we've got a bunch of microbiome data and often a lot of phenotype data. But usually in those data sets, we're lacking host genetic data. And so it's actually really challenging to find data sets where all of these different types of data exist to do this kind of work in a really kind of broad survey type way. So what am I going to do? Where am I going to find this magical data set to do this kind of analysis with? So I turned to the Twins UK cohort, which, as the name implies, is a big cohort of twins that reside in the UK. This is an effort that was started by Tim Spector a couple of decades ago, and they have been tracking twins in the UK for, like I said, a couple of decades. And there's a lot of data, molecular, phenotype, and otherwise, that are available for these individuals. So do the study I'm going to tell you about, I requested a bunch of different phenotype data, most of which was kind of immune-related, because I wanted to focus on things that I thought the microbiome might be affecting. Today I'm going to tell you about just 23 different traits, 10 of which are what I call binary phenotypes. It's a disease you either are diagnosed with having or not. So these are things like asthma, allergies, eczema, that kind of thing. And 13 different quantitative phenotypes. So these are things like body mass index, height, weight. We've got some vitamin levels, that kind of thing. For the microbiome data, we have 16S rRNA gene sequencing for these samples, which is what I'll be talking about today, for about 3,000 stool samples in this study. This was collected by a former graduate student in my postdoc lab, Julia Goodrich, who's fantastic. And then for genetic data, we have imputed SNP chip data. So we have something like 1.3 million variants all across the genome. So using all of these data sets, I'm going to kind of try to combine these all together to get at this question of how much is explained by genetics and how much of the variability in each of these different phenotypes is explained by the microbiome. So, bird's eye view of the approach, in case you don't want to think about the nitty-gritty on a Friday morning at the end of a conference. So even if you ignore the next few slides I'm going to go into, as long as you keep this in your mind, this should be fine. So basically all I'm going to do is I'm going to model each of my phenotypes, and I'm going to expand on what is typically thought of as a GWAS null model. So I'm simply modeling my phenotype as a function of whatever covariates might be relevant to include, and also a term that accounts for the relatedness among the individuals in my sample. And by having this relatedness term, this allows me to calculate something that's called percent variance explained that is due to genetics. And simply all I'm going to do is expand this model to include a term for the microbiome. So again, this is all you have to remember. I'm just modeling my phenotype as genetics and the microbiome. In case you want to know the underlying math, here's what this actually looks like in practice. So like I said, we're modeling each of our different phenotypes. We include the intercept term, which is like the average of the phenotype, if you want to think about it that way. We can include any kind of fixed effects that might be covariates that would be relevant, so things like sex, age, if they're on medications, that kind of thing. And then we have this term here, which is a random effect in our model for this genetic relatedness. And how this is actually encoded is this is encoded as a matrix, an n by n matrix, where for each pair of individuals, we have a value for how similar their genetic makeup is. So of course, you are most similar to yourself. You have a relatedness of 1 to yourself. And then any siblings or parent offspring pairs in our data set is something like 0.5. And then unrelated individuals, will share some segments of their genome just by chance. And so by including this n by n matrix, again, this allows us to calculate this variance component of this term. And this we can kind of think of as a somewhat conservative estimate of heritability. It's called chip heritability in the literature. So what are we going to do with the microbiome? How are we going to include this? So one of the things that always struck me as someone who bridges these two fields is that we have something in the microbiome world that is very, very similar to relatedness, at least in structure, which is something called beta diversity. So with beta diversity, you can think of it the exact same way as relatedness, with every pair of individuals, if you are more similar, you show a lower beta diversity. So how this works is, let's say you take person A and person B here. They have fairly different microbiomes, and so we would say that these two individuals have high beta diversity. On the flip side, person A and person C here have quite similar microbiomes, and we would say that these individuals have low beta diversity. And there's a bunch of different beta diversity metrics, because you can weight the abundance of your microbes and the overlap, and you can take into account phylogenetics and all sorts of different things. But in general, this is how this works. And very conveniently, this is, of course, encoded in this n by n matrix, where again, for every pair of individuals, we now can look and see the similarity in microbiome composition across all of the individuals in our data sets. So I'm simply including this additional random effect term here, and this allows me to calculate the variance component of the microbiome in addition to this variance component of this genetic relatedness matrix. So first things first, I wanted to make sure everything was set up well and made sure everything made sense. So I first ran this normal GWAS null model on all of my data just to make sure everything was working as expected. And so here I'm showing you the bird's eye view of all of the different complex diseases in my data set. For the rest of my talk, anytime I'm talking about the genetic component, it's going to be in blue. Anytime I'm talking about the microbiome, it's going to be in orange. And then anything that's left unexplained is in gray. So as you can see, just by eyeballing this, you can see that for most of our traits, we've got some evidence of a genetic component, which is not surprising. The error bars on this are quite large. By human GWAS standards, this is a tiny little baby GWAS, right? We've got 2,000 samples. It is nothing. People are doing GWAS with hundreds of thousands of individuals right now. But this is the type of data that we have when we want to intersect that microbiome data with it. So this all makes sense. But I wanted to confirm that this tracked with what we know from the literature. So I pulled estimates of heritability from the literature for as many traits as I could find. I also have twins in my data set, so I can calculate twins-based heritability on all of my traits. I've put those up in little blue triangles and diamonds here. And then I also calculated this percent variance explained due to genetics or chip heritability using my model. There are now way too many points on this plot, and you can't make sense of anything, so let me throw up some arrows. And what you can see is that this percent variance explained tends to be a little bit lower than what we see with kind of classic heritability measurements. This is completely expected. This is known behavior of chip heritability in the literature. We're only assaying a certain number of variants on our chips. We can only measure common variation, and so we tend to see conservative estimates of heritability. So it's working as expected, but do keep in the back of your mind that our estimates of heritability are somewhat reduced from like full heritability you might calculate in a twin pair, for example. Okay, so what happens when we throw the microbiome into this equation now? So here, bird's eye view, again, the microbiome is in orange with genetics in blue. You can see that in general, genetics explains more of the variation than the microbiome does. But let's zoom in on this microbial component. So here, looking across all of these traits, you can see that there's variability, right? For some of these traits, the microbiome can explain up to 25% of the variability we see in the twins UK. And for other traits, it explains nothing at all. Reassuringly, there was a study by Rothschild and Weisbrod back in 2018. They had a different method, and they were able to use that method to look at things like height and body mass index. And we see very similar estimates to what they calculated for those two traits, which is really reassuring. Additionally, here we have more traits, and this allows us to see that, in fact, it's not just that like we're always going to find some component explained by the microbiome. There really is variability across traits, which makes sense. So back to this broader goal, for complex traits in humans, how much is explained by genetics versus the microbiome? Genetics, we see, explains anywhere from 0 to 80% as expected, and the microbiome explains anywhere from 0 to 25%. It just depends on the trait. So I think now's a good time to take a pause and be very careful about the language and point out that I've tried to be careful about the language. So I keep trying to say things like it's explained by the microbiome, it's explained by genetics. So you might see these results, and the first instinct might be like, oh, okay, so the microbiome is causally contributing to these phenotypes. That may be true, right? There are certainly some really elegant studies in model organisms where we know if you put in a microbiome into a germ-free mouse, for example, if you put in a microbiome from a higher body mass index individual, those mice tend to gain weight more than a mouse that gets a microbiome from a more lean individual, for example. We know that there are causal effects of the microbiome in some circumstances. However, this is a statistical association, right? We are detecting a statistical association between this microbiome term and our phenotype of interest. And what I actually think is more likely happening here is that the microbiome we know is influenced by a lot of environmental factors, and we are probably picking up on those environmentals secondhand through this term. So for example, the diet you eat, the drugs you take, all of that kind of stuff influences your microbiome, and that composition is what we're technically putting in the model, but we're actually capturing that variability in your environment instead of directly microbiome causal effects. And at first, that might seem a little unsatisfactory, like, oh, well, you can't actually say it's the microbiome, and that's true. But I think with that in mind, this actually gives us a really unique opportunity to leverage the microbiome and use it in a kind of what I think is a really cool way. And that's actually to use it as a proxy for the environment. So we know the environment is really important to a lot of complex traits, right? But it's really hard to measure the environment, right? You can only give so many surveys to people to fill out. You can only have them wear so many wearables. And when you try to model that all together, that also gets really complicated, right? So if we can actually think of one metric to use that is a proxy, as a broad proxy for a bunch of different environmental influences, perhaps this could actually be a really powerful approach to account for environment. And so with that said, I was thinking, well, can we actually use this as a proxy of environment? And if we do that, can we actually improve our ability to detect genetic associations with a trait? So basically using this as a covariate in a GWAS to account for all of these environmental factors that perhaps are also important for trait variation. So the kind of ideal world is that we go from having a GWAS where we don't really have any signal, but now when we include the microbiome, we've accounted for that environmental variability, and we now can detect genetic associations with our trait. So how this works in principle, right? With the GWAS, we have individual variants all along the genome. For any particular variant right along the genome, we probably don't expect there to be an association with our complex trait of interest. So we might see an association like this, where if we stratify our phenotype by genotype class, there's no real association. What we are looking for in a GWAS is something that looks like this, where if you stratify our phenotype now by genotype class, we see that there are differences with individuals having more A's, having higher whatever this phenotype is. And what I'm proposing is that by putting the microbiome in there as a covariate, what we're actually doing is we're capturing some of that environmental variability. So we're not necessarily changing the beta on this or the effect size on this, but we're simply lowering the variance, and this allows us to detect this more easily in our models. So how does this actually work in practice? I'm taking that GWAS null model that I just showed you, and I'm simply adding a fixed effect term for each of our different SNPs. And then I can test this model where I either include the microbiome or I don't, and see if there's an improvement in fit. So I ran this for all of the 23 different traits for all 1.3 million variants across the genome, and obviously that is far too much data to show you here. So I'll kind of give you an overview first, and then I'll zoom into one example. So looking across all of these diseases, when I include the microbiome as a covariate, it doesn't help me always. And where I do see improvement are in these traits right here. Reassuringly, these are mostly the same traits where we see a pretty high level of variability of those traits explained by the microbiome, which makes sense. If the microbiome isn't explaining anything, you can throw it in the model. It's not going to change anything. But here, for example, with body mass index, which I'll show you in a second, it's explaining up to 25% of the variation. And what I see is that for my top SNPs in my GWAS, it does improve our p-value a little bit. So like I said, I'm going to show you one example, and this is the GWAS for body mass index. I will preface by saying this is the best example, but the trends hold across other phenotypes as well. So I'm showing you here a Manhattan plot, right? Each point on this plot is a different variant in the host genome, so there's 1.3 million points on this plot. They're ordered along the x-axis by chromosomal position, and then the significance of association is along the y-axis. With the higher up the point is, the more significantly it's associated with the complex trait. So what you can see here, when we run a classic GWAS without including the microbiome, we don't see anything that reaches genome-wide significance. This is, again, not surprising because we have our little tiny baby GWAS of only 2,000 individuals. Of course, we're not going to find anything genome-wide significant. But for body mass index, actually what happens when we include the microbiome is that this variant that's over here on chromosome 3 actually pops over the Bonferroni significance threshold after we include the microbiome. And it does for a couple of these other top SNPs as well. So this variant that I'm highlighting here is an intergenic variant, as most GWAS SNPs are, but it's really close to this EPH-A3 gene that's important in neuronal development. Reassuringly, this SNP has previously been implicated in body mass index GWAS. So we're not finding anything new here, which I think is actually a good thing because we're not finding things that aren't real. We're finding something that people have known about before. We're just, again, improving that power to detect this association with a smaller sample set. Additionally, this variant has also been implicated in waist-hip ratio GWAS as well. So naturally, I first got these results. I was very excited. But then the next step was, is this real? Or is this a statistical artifact because I'm introducing more covariates into my model? So I did a bunch of testing to try to rule out that possibility. And the most convincing test that I did was this permutation test for beta diversity. So I wanted to make sure that I was still including the same number of terms in my model, but I wanted to see if I actually split apart the biological link between beta diversity and our genotypes or our phenotypes. Am I actually preserving this signal, or does this go away? So what I did is I permuted those beta diversity measurements across all pairs of my individuals, and I looked to see if we saw this improvement in GWAS performance that I saw with the real data. So here I'm showing you the actual p-values from the two GWAS. So this blue one is from the normal GWAS. This orange point is the GWAS that includes the microbiome. And so you can see that improvement in performance here for this top SNP. And now in gray, I'm showing you the results of 1,000 permutations where I've permuted this beta diversity metric. Of course, in the GWAS where I don't include beta diversity, that doesn't do anything, doesn't change anything. However, in the GWAS where I do include beta diversity, you can see that, of course, there is some noise around what is the null estimate here without including the microbiome. However, our actual improvement in p-value is much outside of any of these 1,000 permutations. So this is statistically significant, and this is the case for all of these top SNPs that I identified. I didn't identify a single case where it seemed like this was within the kind of normal bounds of statistical noise that we might expect. So I think this is a real effect where by including the microbiome, perhaps we are mitigating the effect of environmental variance, and this allows us to detect these genetic associations more easily. I will also say this doesn't work across the board. So I told you there's a bunch of traits where we don't see any improvement at all, but even within GWAS, within the body mass index GWAS, for example, it's not like every single SNP jumps up a bit. It seems to be that this is a SNP and microbiome kind of interaction effect, whereby for some SNPs, this relates to an improvement, and for other SNPs, it doesn't relate to any improvement. And so I think this offers the potential, actually, of using this to look at gene by environment interactions, which are something that are of major interest to the field, but are incredibly hard to pin down because you have to do so many tests to find them. So I think this is one way we could also prioritize looking at these gene by environment variants, which I think is really exciting. So to wrap up, a couple of things I hope you walk away with. Number one is that the microbiome, I will put in air quotes, can explain up to 25% of the variance that we see in these traits in the twins UK. One of the things you might have noticed is that most of the traits that actually showed kind of bigger variance explained were not the ones I anticipated were going to pop out. I requested all these immune traits thinking that's what was going to pop out, but actually it was things like body mass index and vitamin levels that are circulating, things like that, where I actually saw the kind of most explained stuff. So I'd be really interested in using this on more phenotypes and in more populations to see how well this translates. Also, including the microbiome can improve our power to detect genetic associations with disease. And I hope the message that this sends is that we can get more of the human GWAS people to be collecting microbiome samples, because I think it can help their studies, and vice versa. I hope the microbiome people can also start to think about collecting genetic data, because I think that there's a lot of potential by integrating these two data types together. So with that said, this is definitely work I couldn't have done alone. I started this when I was a postdoc with Andy Clark and Ruth Lay, and I've continued this work in my lab now at Penn State with all of these folks. So thank you so much for your attention, and I'm happy to answer any questions you might have.